Good day. My name is Kent Armstrong, president of TerraStryke Products, LLC. Here to discuss with you leveraging of microbial behaviors to realize contaminant destruction via biostimulation alone. We at TerraStryke develop additives designed to enhance the indigenous population, microbial populations in the ground, and to achieve greater performance with respect to contaminant degradation. What is not biostimulation is high impact, high cost, intensely aggressive activities, which have benefits, but also they have their disadvantages, mostly in large terms in cost. What is biostimulation? Is a very passive, non-invasive approach that allows nature to address and utilize the contaminant energy source that's present the way nature intended. Cost effectively, minimize the impacts, maximize the performance. At TerraStryke, we recognize that the treatment zone is more than just a contaminated area. It's an ecosystem of indigenous microbial populations. That's who we're trying to help. But first, we need to enhance the nutritive capacity of the treatment zone to support those indigenous bacteria. Under the current conditions at a treatment zone that's contaminated with, say, chlorinated solvents or petroleum hydrocarbons or other, the mass amount of contaminants has overwhelmed the system. So that's an ecosystem that's under stress. And when it's under stress, the indigenous microbes do not have the capabilities to grow, to develop, and communicate like nature wants them to when they have a healthy system. And so what the purpose of the biostimulation additive is to restore the nutritive capacity of the ecosystem to effectively eliminate environmental stresses. In doing so, we allow the microbes to work collectively and establish what we call biofilms. Biofilms are protective housing where they will reside and share genetic information at rates 10, 100, 1,000 times faster than they would in the bulk water surrounding them, and allows them to evolve in real time faster to the conditions around them. In doing so, they achieve superior levels of sustainability, and we see greater levels of contaminant destruction with minimal impacts and low costs. Power of unicellular organism has become, over the last 20, 30 years, quite apparent. Years ago, we thought that the basically prokaryotic microbes were in general loners, capable of very little, just being a cell with a single strand of DNA. Solitary, and we looked at the planktonics, and in our industry we still focus on the planktonics, the swimmers, as being the key to the process. But we argue with bioremediation 4.0 there's a next step that we can take those planktonic swimmers to. Excuse me, we now know that less than 1% of bacteria in nature exist as a swimming form, as a planktonic bacteria. Over 99% of the microbial populations on the planet live in biofilm. Within a biofilm, they communicate, they talk, they actually signal using autoinducer signals and others so that they can share information, direct phenotypic changes, and in essence, recruit and teach one another how to utilize and exploit the available energy around them that we call contamination. If you get them healthy and if they're able to communicate under a healthy ecosystem, they will determine what benefits the population as a whole. They're able to take census. They're able to see who is around them, what the conditions are around them, and in doing so, work together to achieve maximum performance. If they reach a quorum density, a quorum level, if the planktonic microbes reach a certain level and the nutritive capacity is complete, they'll abandon the swimming roles, they'll abandon the individual genetic roles as a swimmer and assume those in a biofilm, assume a new role that benefits the collective. With an enhanced nutritive system, microbes can establish structures, the biofilm, and within these structures, they create water transport systems where water actually moves 10 to 100 times faster than it does in the bulk water surrounding them. They create nutrient sinks where they can maintain and create sustainability of available nutrients and metabolic needs. They create systems to recycle and remove waste, transporting it out of the biofilm so it doesn't interfere and stall performance. And they maximize the utilization of contaminant energy, the protons of a petroleum hydrocarbon, the electrons of a chlorinated solvent, an alkane or an alkene, and possibly even, we're quite confident, the fluorinated compounds. The power of the unicellular organism means to you with bioremediation 4.0 that you'll achieve superior, more sustainable contaminant destruction with less effort. You'll eliminate the above ground energy consuming support equipment. You'll eliminate the nuisance noise, the vapors, the emissions, the costs, the permitting associated with that above ground equipment. You'll enhance the remediation process by not creating greenhouse gases, but rather you'll be sequestering greenhouse gases throughout the entire process. You'll realize remediation objectives with less impacts and less cost, simply by leveraging 4.5 billion years of nature's experience. Biofilm developed because it's the way that they provide protection and develop and evolve so that they can maximize energy utilization. This is how the planktonics got out of the primordial soup 4.5 billion years ago and started building themselves into individual species within biofilms, which subsequently established the development of multicellular organisms. What we're doing on our level is developing a biofilm, which in essence acts as a multicellular organism rather than an individual planktonic organism. With this slide here, we see that the biofilm develops first by, yes, enhancing the number of planktonic microorganisms present. We have to reach a quorum level. We have to reach a density such that the autoinducer signals being emitted reach a concentration that tells the microbes present to collectively change their phenotype, become sessile and adhere to a matrix, grow, split, and all the time secrete polysaccharides, peptides, and other metabolic substances that make this gelatinous goo, this upper right-hand corner, which is a mature biofilm in which they can do incredible things. The picture down the bottom right is an artist rendition of what a biofilm may look like. They're heterogeneous. They come in all shapes and sizes. They're single species, they're multi-species. It depends on the condition and what's available, they will make it. This shows the water transport, the water channel systems within the biofilm. And it also shows scouts leaving the biofilm after having lived within it, worked within it, shared genetic information within it, so that these scouts, these newly distributing planktonic forms coming out of the biofilm, are going to be able to show complete different genetic expressions, more evolved, more adapted to the utilization of the protons or the electrons present as a result of the contamination. Biofilm development has been around forever. 30, 40, 50 years ago, we used to think the bottom of the ocean was sterile. There was no life down there. And then we made these really cool submarines, and we got 15, 20,000 feet below the sea. And we saw these flumes, these thermal flumes full of nutrients. And wherever they erupted, you saw teams of life surrounding the cones. Biofilm. Biofilm is such that microbes don't die. Yeah, you put them in an autoclave, you're going to kill them. Yeah, you can pour bleach all over them, you're going to kill them. But in nature, they typically don't die. They assume a starvation mode when things get tough. And as this starvation mode, they're called an ultramicrobacteria. It's basically a shell and a single strand of DNA. There's millions and millions of years of single strands of DNA layered in the soil, layered in the ice, laying in wait for the ecosystem that they are in to be re-enhanced. The development of a biofilm allows enhanced cell-to-cell communication. Biofilm serves as a reservoir of nutrients, energy, and metabolic substances to provide sustainability and protection. The protection keeps them from predation. Amoebas just slide over these things. It can't eat the bugs inside, but it woofs on the ones that are swimming about them as a planktonic swimmer. Those scouts are going into a hazardous, dangerous world. It also protects them against temperature, pH, and salinity. We've seen complete biotransformation of TCE in Saudi Arabia at a pH of 10. We've seen of chlorinated alkanes in Saudi Arabia at a pH of 10. We have TCE complete biotransformation at a pH at times of 3.86 in Ohio. We've also demonstrated in the lab complete biotransformation of PCE in 3% salt water. And we're working to replicate that now and look forward to sharing that information. As I said before, biofilms are heterogeneous. They consist of multi-species cultures, but they're found everywhere in nature. Biofilm is the plaque in your teeth. Biofilm is the infection on your hip implant. Biofilm is the infection on your stent. Biofilm is cystic fibrosis. The benefit and the hazard of biofilm is it's so misunderstandable. It's wicked complicated because they can share genetic information thousands of times faster than they can in the bulk water. We know what swimmers can do. We're at a loss still of what they're actually doing inside. More and more research is being performed. We hope to spearhead that, and we are spearheading that within our industry. And there's a lot of fun stuff to learn. But in terms of practical knowledge, biofilm outperforms without any adverse conditions. Two of our flagship products I'm going to talk about today, one being TPH Enhance, which is designed to stimulate heterotrophic petrophilic bacteria passively to restore their nutritive capacity of the ecosystem. I'm going to show you an example of a site in Seattle where the consultant uses minimal deployment of additive. He's using 200 pounds of TPH Enhance with about 400 gallons of water. It's a 1-to-2 mixture. He's deploying that 400 gallons of additive solution 3 times. He did once in March of 2021, again in April of 2021, and then in May of 2021. You can see at the initial deployment, the small arrow here, then we have 2 subsequent deployments. After the initial deployment, the line in green represents heterotrophic plate counts. Yes, heterotrophic plate count is an important metric. Just because it's only $45 doesn't mean it's not important. You need to know the densities. Here we see you might not have to know the names. You need to know the densities. The genes are there. We have a 4.10⁻⁴ baseline, and then as soon of colonies per milliliter. And then right immediately after he introduces the additive, within a short month, we see an increase of 2.8 times 10⁴. A significant increase in the densities of bulk water swimmers. Soon thereafter, a time at which we believe we had reached quorum densities being this dotted line here, where we believe the concentration of auto-inducing signals reach a level sufficient to indicate to all the swimmers there to, hey, stop swimming. Phenotypically becomes sessile. And within another month, we see a drop in that same heterotrophic plate count, that same population of swimming bacteria go to below detection levels. After the second deployment event, we still don't really see much change in the dissolved phase contaminant concentrations, but they knew there was lots of saturated sorbed mass. We got oil range organics and we got diesel range organics at this site. After the second deployment, the biofilm has established itself. They secrete biosurfactants. They're secreting their polysaccharides, peptides. All of these enhance the washing of that residual Zor mass into the dissolve phase where it's available for them to utilize. They can't utilize it when it's stuck to the soil. And what do we see as this is occurring? Boom, we see a massive increase in dissolve phase concentrations. And I'll tell you, I agree. I'll admit, when I saw this first set of data, it was like, oh my goodness. Come on, bugs, let's go. We saw an increase from 0 to 100 micrograms per liter oil and diesel range organics to 300,000 and 400,000 respectively within a month. And all this time, the heterotrophic plate count, the swimming bacteria densities, were recorded below detection limits. Soon after the third deployment event, this enhanced microbial population in this bioreactor within this treatment zone crushed the available protons, destroyed, assimilated the available petroleum hydrocarbons, such that within the next month, that 304,000 and 400,000 micrograms per liter went to 8,100 micrograms per liter diesel and below detection levels on the oil range organics. Only then did we then see the return of the planktonic swimmers. But not only did we see a return of the swimmers, we saw a return of the swimmers to almost 3 times the level that we saw when we assume we had reached a quorum density of 2.8 times 10 to the 4. We're at 800,000 colonies per microliter now, per milliliter right now. And then a couple months later, they reached 10 million colonies per microliter. And these are educated microbes. These have lived in the biofilm. These have exchanged genetic information. These have been recruited. These have been shared. These have been taught. So now you have not only do you have massive amounts more available to do their job, they're also educated. Their genetic expression is going to be completely different and more adapted to the system where they're living right now than these were when we started this process because of that experience in the biofilm. At another site in Seattle, the same consultant utilizing the same process of 200 pounds of additive to 400 gallons of water, creating an additive solution of about 400 gallons. He puts it in slowly 3 times. The events were 3 times, once in April of 2018, once in July of 2018, and once again, in March of 2019. June, January, and March. What we see here is with the green line is the immediately after additive deployment, we see an increase of additive availability in the groundwater, represented by increased concentrations of TPH enhanced. These pink bars represent relative concentrations of the microbial populations, heterotrophic plate counts. We had a small population existing of swimmers, and as soon as we enhanced the area, we saw that concentration doubled, if not tripled, in terms of densities. At the same time, as the concentrations, as the densities of microbial populations were increasing, they were enhancing the degradation of the gasoline range organics and either solubilizing or generating as a result of the degradation of the longer chain hydrocarbons. We're seeing an increase in BTEX concentrations as the populations are growing. At peak population, we see peak solubilization. We see a rapid period of massive solubilization of the residual mass sorbed to the saturated soils, and then a rapid depletion of additive availability, such that, let me get rid of that thing. Such that we see the rapid depletion of additive availability. At the same time, a steady decrease in the groundwater dissolved phase concentrations from peak bioavailability is occurring. Also, the heterotrophic plate counts during this period over 6, 7, 8 months are basically below detection levels. They disappeared again. Because right here, we reached a quorum density. They formed a biofilm. They went away. They stopped swimming. So when you grabbed your groundwater sample, they weren't there. They're in the biofilm working collectively to degrade over 8, 9 months the available protons to attain a 94% reduction in gasoline range organics and a 98% reduction in BTEX. Only after that happens, only after the massive amount of protons have been destroyed, utilized, do we see a return of the planktonic concentrations such that the densities increase to at or above previous quorum levels. And this was in a source zone, so we had small amounts of beginning population, increases and then disappears. Source zone is more toxic. Just down gradient from that area at monitoring wells W5 does the same thing with a couple injections. You just have one injection event. And because this area is outside the source zone, not as toxic, doesn't have Zorb mass, we say high concentrations of dissolved phase contaminant, and we see a healthy population because the nutritive capacity hasn't been completely scavenged. They're living, but they haven't yet reached a quorum density. As soon as we increase the presence of additive, you can see the green line goes up. Concurrently, the population's densities increases as planktonics. The dissolved phase concentrations in 2 months decrease 99% each. But the heterotrophic plate count, the density of the bulk water planktonic swimmers, again disappears. Later, after all the protons and contaminants gone, we see the swimmers reappear. They have a period of advection into the treatment zone. We see them disappear again. We see some degradation, and then all of a sudden, boom, a massive release of planktonic swimmers from this biofilm after having degraded over a 9-month period 85 and 95% of the contaminants present. We can deploy this stuff in any way. It comes dry, 33-pound bags. You mix it with site water, gravity feed, direct push, infiltration gallery, any way you want to put it in, we can get this stuff into the ground. You blend it 1-to-2 with water, 1-to-1 with water. It's so thin a driller's pump won't carry it, but it has maximum enhanced distribution in the subsurface. And we typically do our deployments at 15 to 25 foot intervals if you do a direct push. We also work compatible with any other remediation program out there. If you have a carbon or a abiotic process at a site where it's reached its capacity, we'll reinvigorate that capacity and complete your job. If you have a thermal treatment system ongoing and a fairly speedy groundwater system, we'll create a bacterial halo around that system, and you won't lose that dissolved phase contaminant down gradient. We'll utilize it. If you want to do a phytoremediation program, not only will we enhance those microbials, but we'll enhance those plants that are working as well and create an incredible rhizomal biofilm to create sustainable degradation of whatever contaminant you want, like you've never seen before. Where do we work? We'll look at any site, but we will be the first ones to tell you if our additives are not appropriate. They are majoritively appropriate, but there are sites where it doesn't work. There are sites where it can't be deployed. You always remember with bio, this is a contact sport. It's got to get there. The beauty of our additives are their thinness and their ability to distribute in addition to their power. Our other flagship item is ERD Enhance. This is our biostimulation for chlorinated solvents. This one works dramatically well such that I can give you reducing conditions, sustainable reducing conditions after a single deployment event that will give you last for decades, 1 to 2 decades easy. We had a site, a friend of mine has several Superfund sites throughout the Midwest. Had a fractured limestone bedrock site where tons of pump and treat is ongoing. Baseline total CVOCs were about 28,000 milligrams per liter. There were no daughter products there, meaning you had nothing but parent. This is basically all TCE. The CVOC total is all TCE, 28,000 of it. There's no biotic activity going on. They ran this system for over 25 years, removing billions of gallons of water, and yes, removing tens to thousands of pounds of contaminant, but at quite a cost. They turned the system off in December of 2017, and yes, the system went right back to baseline, 28,000 micrograms per liter, milligrams per liter, total CVOCs, and a parent-daughter ratio of 100%, all parent. And this was a probe they pulled out 1 month after deploying 33 pounds of ERD Enhance with 50 gallons of water. We used to have pulled that out and said, oh my gosh, you fouled your well. But this is anaerobic. Fouling a well is under aerobic conditions. That is a biofilm. And what that did within 12 months was allow the microbial populations present to destroy that CVOC contaminant all the way down to only 15 micrograms per liter of vinyl chloride was remaining 12 months later. The parent-daughter ratio went from 100% to 0%. There was no parent left. We saw complete biotransformation in 12 months. Not 25 years. It's been another 2 years or a year and a half since, and we have those exact same conditions at this well. One of the things we did this last summer during the pandemic, during the height of the pandemic, is we had an independent comparative evaluation of ERD Enhance against abiotic process. We did a microcosm study, retained a company to do it. They spiked the microcosm with 50,000 micrograms per liter PCE. We established a groundwater condition similar to an Ohio site where we've been working for 10 years now on various treatability evaluations. And what we saw wasn't a surprise to us, but I believe it was somewhat of a surprise to the performer of this microcosm study. We did see, yes, the abiotic process realized greater dissolved phase reductions in CVOCs. Oh, right here, the abiotic process, you saw a 77% reduction, 76% reduction in dissolved phase contaminants. But that's what they do. If you can get it to contact the mass, it'll absorb it. You saw the same reduction in parent PCE, because again, it doesn't care what molecule it is, it's going to adsorb that molecule. But what is of interest, you saw little to no destruction. You're trapping it. You're hanging on to it. You're stabilizing it, but you're not destroying it. What we did see with ERD Enhance, the organic process, is we saw a 91% reduction in dissolved phase concentrations. We saw take that back. We saw a 42% reduction in dissolved phase concentrations, but we saw a 91% reduction in parent PCE. That means we are destroying parent mass. And we also saw a 90% reduction in parent-daughter ratio, supporting that dissolved phase reduction. We did do a combined effort where we combined a small percentage of the abiotic metal with the ERD enhanced formula and found some very interesting things. In this case, we saw similar reductions, enhanced reductions in total dissolved phase, in parent PCE, basically statistically similar to the original formulation. The same study, we looked at DHC counts, how many dihalococoides were present, and we saw that in our flagship ERD Enhance, we saw a continued rate of increase in DHC population, such that with ERD Enhanced alone, the standalone product, we saw a sustainable increase in DHC throughout the 28-day microcosm process. The control, we saw a decrease actually. And with the abiotic metal, we also saw a decrease. Granted, you're not putting in an abiotic metal to generate biological growth, so that makes total sense. Interesting here, the combined, we saw an initial increase, and then we saw a decrease in DHC populations. We'll talk about that in a little bit. Also, with regards to signaling, let me go back to that last slide. In the ERD enhanced formulation, we have a compound that we believe that be very critical with respect to microbial signaling, with the auto-inducing signaling. And we had one microcosm generated that did not contain that communication compound. And we believe this difference in DHC growth may be representative to the effect of not having that ability to communicate. The communication is measured by looking at the AI, the auto-inducing signal, the concentrations of auto-inducing signals in the groundwater. What we noticed was we saw a sharp decrease in we saw a continual rise in auto-inducers with both ERD enhanced formulations. But earlier on, well into the study, we saw a sharp increase in signaling and then a sharp decrease in signaling with the combined formulation. We also saw that, as you can see in the bottom slide, that the combined formulation also formed the greatest amount of biofilm throughout this microcosm study. And so what we believe is happening here is because they formed that biofilm, sorry for the mistakes there. Because we formed that biofilm, They didn't need to signal anymore. And so the signaling concentrations in the bulk water slowed and decreased in response to the bulk water densities going into the biofilm, where now they're commuting at even faster rates inside the biofilm. We believe the two ERD formulations will in turn, in time, show a similar drop, but it just takes longer. What we now know is by the introduction of a small percentage of abiotic metal with our ERD enhanced process catalyzes biology. We have a site in Burlington, Ontario. This is one of our earliest sites. We were amazed at the data. We were amazed at the results as it happened, and now we know why. Owner inherited it through bankruptcy in early 2000s. It was appraised at 2011 for roughly $700,000. Highly weathered shale, concentrations of PCE, 100 to 180,000 milligrams per liter, well above solubility, lots of residual mass, and highly exceeding MOE standards, Ministry of Environment standards in Canada. They excavated as much as they could with the source zone saturated soils, backfilled the pea gravel, and installed an infiltration gallery where they gravity fed 1,000 pounds of ERD enhanced with 1,000 gallons of water twice. Passively, slowly trickled it in, with allowing gravity to take it as the system would receive it, once in March of 2014 and then again in June of 2014. Within 2 years of the deployment of that additive, 99.4% of PCE was destroyed. 99.9% of TCE was destroyed after a small increase. Cysts increased 3,600% during the process and then decreased greater than 99.99%. Vinyl chloride decreased 99.9% after a slight increase at the end of the second year of evaluation. We had an overall decrease of 99.95%. Total CVOCs. Ethene was generated throughout this entire process, meaning complete biotransformation and contaminant destruction was ongoing. The property was valued 4 years later, in the beginning, at $680,000. And originally it was advised that a pump and treat system would cost them roughly $750,000 and require 12 to 15 years of operation, effectively eliminating the value of the property. He allowed me to do a pilot study. It was successful. We did this remediation program for $223,000. And within 4 years, that site was redeveloped into an active Scottish grocery store, Scottish bakery grocery store, valued in 2018 at $2.5 million, $1 million of which the owner attributes directly to our passive-aggressive remediation program. What will we do for you with Bioremediation 4.0? We'll help you solubilize, liberate low perm and L-NAPL, D-NAPL contaminants. We'll help you expedite the destruction of dissolved phase contaminants, maximize the utilization of the energy available. We make the energy available be utilized as electron donors or acceptors, like nature wants to. You'll achieve sustainable remediation without above ground support equipment and the permitting costs associated with that. And you'll actually sequester greenhouse gases during and after the remediation strategy program. You'll realize site compliance with less impacts, less costs, simply by letting nature have it. Lots of options out there. We have solutions. We look forward to your call and working together to devise a strategy to meet your compliance objectives. If you can go to our website, you'll see that there's a banner on the front page that says Site Remediation Quote Form. Simply click on that. A few minutes, fill out some basic information for us. And within a day or so, maybe sometimes even hours, we'll have a preliminary estimate for you from which we can begin the development of a passive biostimulation strategy to end your environmental compliance liabilities now. Thank you ever so much. Here's our contact information. Don't ever hesitate to call. Don't ever hesitate to email, contact us. We're on LinkedIn. Several videos like this are we actually have some on YouTube. But please call to discuss. We have a solution for you. And this is the next generation of bioremediation. Thank you very much. Be safe. Have a great day. Do no harm.